Arnoud Van Harst has a long-standing experience in clinical pharmacology studies, including drug-drug interactions, and is also a subject matter expert in Celerion's respiratory research team. He supports a broad range of drug development programs at Celerion's clients. Arnoud joined Celerion in 2015 and has over 25 years of drug development experience at biotechnology companies and CROs. He received his PhD from the School of Medicine at Leiden University and qualified as a clinical pharmacologist in the Netherlands. Over to you, Arnoud.
Thank you, Archie. During this webinar, I will first of all share with you how to evaluate the effects of food changes in gastric pH and rate of gastric emptying on oral drug absorption. And subsequent to that, my colleague Sabina will dig into the impact of concomitant GLP-1 agonists. For those of you who are not familiar with Celerion, we are a full-service clinical research organization that entirely focus on clinical pharmacology studies. So we specialize in early phase healthy volunteer studies, including product labeling studies. And in addition to clinical conduct of studies, we offer regulatory and protocol writing services, data management, and we also cover bioanalysis. So we are a global company with clinics both in the US, in Lincoln and Phoenix, and also in Europe, in the UK, in Belfast. And in total, we have a bed capacity of over 600 beds, which makes us one of the largest phase 1 clinics across the world. In addition, we have bioanalytical labs both in the US, in Lincoln, and in Switzerland and Zurich. We have some additional offices to support other services than clinic and bio lab. As alluded to, our focus is on early stage clinical pharmacology studies. And in this graph, you can see an overview of the types of studies that we have conducted ever since 2010, highlighting the over 100 food effect studies that we have conducted and nearly 400 drug-drug interaction studies. And based on this wealth of experience with these types of studies, we're happy to share some of our tips and best practices here today regarding food effect studies and certain drug-drug interaction studies involving acid-reducing agents. So let's first zoom in on food effect studies. Intake of food can interfere with the absorption of orally dosed drugs, similar to drug-drug interactions, which can also establish a change in absorption that subsequently can lead to a change in exposure and have impact on either efficacy of a drug or may generate safety concerns. And there's a couple of mechanisms by which food may actually interfere with drug absorption, and some of them are listed here. So for instance, food generally goes with an increase in gastric pH, and similar to interaction with gastric acid reducing agents, this may change the solubility of a drug at the established higher pH. Moreover, food will lead to a delay in the rate of gastric emptying, which will increase actually the residence time of a drug within the stomach. And food also goes with an increase in bile salts that may sequester dissolved drug molecules. And finally, the lipids from the food may help lipophilic drugs to get better solubilized. Now, it is important to assess food effects early in clinical development so as to improve or optimize the design of later stage studies. FDA guidance for food effects was lastly updated 2 years back in 2022, and it highlights that particularly the effect of a high-fat meal should be assessed in early stage of development, particularly as a pilot. But as high-fat meals are more likely to affect the gastrointestinal physiology and therefore will have a larger effect also on the drug bioavailability. But the updated guidance from FDA also recommends to consider a food effect study with a low-fat meal For instance, if a high-fat meal had a great impact on toxicity or on efficacy of a drug, or in the case that patients cannot tolerate a high-fat diet. And still, a pivotal food effect study may be required at a later stage. For instance, if the to-be marketed formulation differs from the formulation that was tested in the pilot study, or to confirm clinically relevant findings with regard to safety and efficacy. And in terms of the meals that are defined within the guidance, I will show some of the examples we use of meals in one of the case examples hereafter. So food effect studies are usually designed as randomized single dose and two-way crossover, although additional arms may be added, for instance, to accommodate different types of meals, or if multiple formulations are to be tested in the same studies. But in a two-way crossover, one basically has a fasting arm where the drug is administered after an overnight fast, and in the fat arm, a meal is consumed 30 minutes prior to administration of the study drug, but also after an overnight fast first. Water intake is restricted from 1 hour before until 1 hour after the intake of the study drug. And apart from the planned meal as per protocol, food should not be consumed at least up to 4 hours post-dose. Food effect can be assessed in a pilot study, which is usually incorporated into a single ascending dose study and typically tests the high-fat meal only. But it can also be evaluated in a pivotal study, which is usually done as a standalone study, where the sample size is powered. And optionally also an arm can be added, or a part of the study can be added where acid-reducing agents effects are considered as a drug-drug interaction part. In this case study that we conducted, we actually had a 3-arm design, because in this case, the investigational drugs target population was restricted to a low diet fat. And for that reason, the regulatory authorities requested us to include studying the food effect of a low-fat arm. So here we had a fasting arm, and in addition, a low-fat, low-calorie meal. And in the 3rd arm, we tested a high-fat and high-calorie meal. And we enrolled a total of 24 healthy adults that completed the study. As said, the meal was given 30 minutes before the administration of the study drug, and in the table shown on the bottom here, you can see how these meals looked like in practice. So for instance, the low-fat meal would consist of skimmed milk, boiled eggs, and a package of oatmeal, whereas the high-fat meal would be composed of whole milk, fried eggs with bacon, slices of toast, and some hashed brown potatoes. This graph shows the drug concentrations from this case, where the PK in the fasted arm is shown in red, and those for the food effect arms are shown in blue for the low-fat meal, and in green for the high-fat meal. And on the right, you can see the key pharmacokinetic parameters. But it's clear from these data that under fat conditions, both with a low and a high-fat meal, there was a considerable reduction in the maximum concentration of about 25%. And also the overall exposure was reduced by about 15%. In addition to that, you can see that there is a slight decrease in the absorption, as witnessed by the time to reach maximum concentration, which was slightly longer. But while the differences established here were statistically significant, the reduced exposure was considered to be not of any clinical relevance. Now let's switch to drug-drug interactions with drugs that may change gastric pH. FDA issued specific guidance for acid-reducing agents drug-drug interaction studies. And again, these types of studies are recommended early in clinical development, particularly if the patients, the target population, are likely to be taking acid-reducing agents, such as in many oncology indications. The guidance also contains a framework, particularly for weak-based drugs, which are at greatest risk of pH-dependent drug-drug interactions, that helps assess the risk of drug-drug interactions based particularly on their solubility. And when designing acid reduction agent interaction studies, one can choose between different types of study depending on the exact purpose of the study. So for instance, one wants to maybe evaluate the worst case scenario or maybe establish a risk mitigation strategy. So this is the snapshot from the guidance, highlighting the framework to assess the risk potential for interaction with acid-reducing agents, which is essentially based on solubility of the drug at a higher pH. And here the solubility is considered for a maximum therapeutic dose in a volume of 250 mils. And if this really is very different from the pH under a lower pH, then there is a good likeliness that the drug may interact with acid-reducing agents, and an in vivo study may therefore be recommended. Now, gastric pH is regulated by proton pumps that are located in the gastric mucosa, and that secrete hydrogen ions to maintain a low gastric pH. But their activity is also regulated by mediators like histamine. And moreover, several types of acid-reducing agents can be used for the treatment of gastric acid disorders, and these agents may increase gastric pH. So for instance, antacids simply neutralize secreted hydrogen ions, whereas proton pump inhibitors directly inhibit gastric proton pumps and are the most potent suppressors of gastric acid secretion. And finally, there are histamine 2 receptor antagonists that antagonize the histamine-induced proton pump activity at the basolateral side of the mucosa. And changes in gastric pH may lead, particularly for weak-based drugs, in a decrease of the dissolved drug, and thereby also in a decrease of absorption. And particularly if this is a change greater than 25%, then that may be of clinical relevance. For weak acid-based drugs, there's less experience with changes, but in theory, one can imagine that an increase in pH may lead to an increase in the dissolution of weak acid drugs, which may go with certain risks, such as increased numbers of adverse events. In an acid reduction agent interaction study, the timing and duration of agent treatment depends basically on the selected type of agent. So while histamine 2 blockers are administered shortly before the study drug and antacids are dosed concomitantly, proton pump inhibitors require pre-treatment for several days. Proton pump inhibitors have a stronger and longer lasting effect on gastric pH and therefore are also considered to represent the worst case scenario. And if the study establishes a clinically relevant interaction with a proton pump inhibitor for a particular study drug, the label may state that the use together with proton pump inhibitors or H2 blockers should be avoided, and intake of antacids should be staggered. But one can also evaluate alternative design strategies, such as dosing regimens with lower doses of proton pump inhibitors, for instance, or with staggered doses of histamine 2 blockers by means of a mitigation strategy. Acid-reducing agent studies are commonly designed as crossover studies, but certainly when proton pump inhibitors are tested, they follow a fixed sequence. And they usually engage a single dose administration of the study drug that is administered under fasting conditions. And across the population, it's good to notice that there is a high prevalence in general of the Helicobacter pylori, which is known to affect gastric pH. This is a bacteria associated with gastric ulcer disease. So the status of H. pylori should be assessed during screening, as this may be relevant for the outcome of the study. And the study drug and also the acid-reducing agent that is administered should usually be at the maximum recommended therapeutic dose. Now, in this case study of a acid-reducing agent, where we tested a proton pump inhibitors on its effect on the relative bioavailability of a orally dosed antiviral drug. This study was conducted in 14 healthy subjects that all completed the study, and the pharmacokinetics of the drug were assessed over 24 hours to establish the maximum concentration, Cmax, and the overall exposure or area under the curve. Now, this was a fixed sequence where in period 1, first the antiviral drug was dosed a single dose and followed by pharmacokinetic sampling. And in the second period, subjects were pre-treated with the proton pump inhibitor rabeprazole for 5 days, and on the 5th day, the antiviral drug was tested. And again, pharmacokinetic samples were taken over 24 hours. And on this slide, you can see that the effect of the proton pump inhibitor on the study drug was obvious. So here on the right, you can see that the drug alone, as shown by the red curve, had a much higher peak than when the drug was given after pretreatment with the proton pump inhibitors. So the Cmax decreased by over 50%, whilst the total exposure of the first 24 hours decreased by about 30%. And in addition, the half-life of the drug was prolonged from 6 hours to 12 hours. Now, as alluded to, acid-reducing agents interactions are particularly relevant in oncology, since many cancer patients take these types of drugs to help mitigate the gastrointestinal side effects of certain drugs like tyrosine kinase inhibitors. And this graph shows the prevalence of use of acid-reducing agents by cancer type. And actually, the prevalence ranges from 20 up to 70%, with gastrointestinal and pancreatic cancers having the most widespread co-administration of acid-reducing agents. And the relevance of that is illustrated by this study taken from literature, where the impact of an acid-reducing agent was shown to be immense, in this case for the tyrosine kinase inhibitors, dasatinib. The overall exposure to dasatinib was strongly reduced after pretreatment with a proton pump inhibitors, as you can see by the green curve, with a 80% reduction of the overall exposure or area under the curve. And the effect of in this study also, the effect was shown that of a acidifying agent being able to restore the low gastric pH and thereby abolish the effect of co-administration with the proton pump inhibitor. So the blue curve shows the dasatinib drug alone, and the red curve shows how the pharmacokinetics after using an acidifying agent got restored. So clearly, reducing agents may have a strong effect on drug absorption. And finally, I want to touch upon drug-drug interactions due to changes in gastric emptying. As said, food intake can prolong gastric residence time, and any such change in gastric emptying can thereby also affect the absorption of a particular drug. But likewise, certain drugs may also change the rate of gastric emptying. And you may be familiar with prokinetic agents that can actually speed up the gastric emptying rate. So for instance, cisapride and domperidone may increase gastric emptying rate and, for instance, lead to a much faster time, shorter time to reach the maximum concentration or Tmax. On the other hand, there are drugs that may delay gastric emptying, such as clozapine. And as Sabina will also highlight hereafter, GLP-1 agonists like exenatide have a strong reputation of delaying gastric emptying. Now, there are several ways to assess gastric emptying, and in the clinic, the golden standard used for clinical diagnosis, like assessing whether someone has gastroparesis, is gastric emptying scintigraphy, in which a radiolabeled meal is used to assess physiological functions of the stomach using imaging techniques. A less burdening study for healthy volunteers would be the use of a non-labeled meal with using 13C labeled octanoic acid. And after consumption of that meal, a breath test can be applied to measure the exhalation of labeled CO2 to assess the rate of gastric emptying. And another technology is the use of a smart pill wireless motility capsule system that is also FDA approved, which helps a measurement of the transit of a pill in the stomach, the small intestine, and the colon, and thereby detecting different or changes in pH. While this is a method that we have applied in the past, unfortunately, the smart pill has been taken off market over the past year. Now, finally, a very elegant method to assess gastric emptying that is very useful in clinical drug development is a paracetamol absorption test, where the pharmacokinetics of a tablet of paracetamol is taken as a surrogate measure of gastric emptying. And Sabina hereafter will dive into more details in an example for that. So in summary, food intake can interfere with oral drug absorption, and also gastric pH changes, for instance, due to concomitant use of acid-reducing agents, may affect drug absorption. And likewise, the rate of gastric emptying may affect drug absorption too. So these types of changes established by food, acid-reducing agents, and gastric emptying rate-induced changes may all impact drug absorption and consequently also have consequences for safety or efficacy of orally taken drugs. And with that, I want to hand over to Sabina.
Sabina serves as Celerion subject matter on metabolic diseases, supporting early drug development of weight loss products like GLP-1 agonists. She joined Celerion in 2015 and has held several roles with increasing responsibilities. Sabina obtained a PhD in biochemistry from McGill University in Canada, and her training includes 3 postdoctoral training. I'll hand over to Sabina.
Thank you for that introduction, Isaac. Now I'd like to take the next few minutes to discuss drug interactions in the era of GLP-1 receptor agonists. Now, GLP-1 drugs have been making headline news as the blockbuster drug could not only impact healthcare, but other sectors as well. Here are a few snippets from news outlets highlighting how the weight loss drug could impact the economy. And feeding into this hype is the growing demand for GLP-1 therapies. So over the past 6 years, there's been an increase by 700% in GLP-1 prescriptions. And according to the World Health Organization, 1 in 8 are living with obesity worldwide. So the number of patients on a GLP-1 drug could potentially increase. And then researchers are also looking into other indications as well. So what I'd like to do is take a forward-looking approach to discuss how GLP-1 therapies could impact new drugs in development. Before we begin, let's discuss GLP-1. It stands for glucagon-like peptide. It's an incretin hormone secreted from the intestinal cells upon food intake. GLP-1 analogs have been shown to control glucose as well as reduce body weight. They are indicated for the treatment of type 2 diabetes and obesity, and can improve cardiovascular morbidity and mortality. Now, exenatide is a naturally occurring peptide that shares homology with GLP-1, the native human GLP-1. It was first identified in the saliva of Gila monsters. And exenatide is the synthetic version, which was the first GLP-1 drug to hit the market. Now we have several GLP-1 receptor agonists available. Some share a backbone, the native human GLP-1 backbone, while others like exenatide have an exenatide backbone. There are also dual receptor GLP-1 and GIP analogs like tirzepatide. And then the next generation of these drugs may include triple GLP-1, GIP, and glucagon receptor agonists. So GLP-1 controls glucose through insulin sensitizing effects. In the pancreas, it will stimulate insulin secretion. It then also promotes glucose uptake within skeletal muscle and adipose tissue. It has glucose production suppression effects in the liver. In the heart, it promotes cardioprotection and cardiac output. And then in the brain, works to control body weight through a decrease in food intake through satiety signaling. For the stomach, GLP-1s have been shown to delay gastric emptying. And as we heard in the last presentation, that this could have a clinically significant effect for co-administered oral medications. Now, here's a summary of the PK parameters for several of the GLP-1 drugs available on the market, showing the indication and the year that they were approved, as well as the dose regimen. Now, the native human GLP-1 is rapidly absorbed and reaches a Tmax within minutes. So these analogs have been formulated to extend the Tmax. Short-acting GLP-1s, Tmax is achieved within 2 to 3 hours, whereas for long-acting GLP-1 drugs, the Tmax could be up to 5 days. In general, the GLP-1 drugs tend to have low volume distribution. They are selective for the GLP-1 receptor and have a specific affinity for pancreatic cells. Because they are peptides, there is no CYP metabolism. However, they are cleaved by proteolytic enzymes like peptidases and endopeptidases. And then the peptide remnants can be excreted through renal elimination. Now, there are currently over 200 GLP-1 drugs in various stages of drug development. Shown here, quite a few are innovator drugs, so they could be maybe me-toos or even potentially going for a bio-better. There are also a number of generic GLP-1s and then combination products as well. So if you are developing a novel GLP-1 product, then it's important to consider the oral medications that will be co-administered because of the potential for delayed gastric emptying. Also, if you are developing a small molecule intended for this patient population, it will likely be given along with a GLP-1 receptor agonist. So that leads to the potential for both PK and PD drug interactions. And in terms of PK effects, the exposure of the co-administered drug, if it is increased, could potentially result in adverse effects. A decreased exposure could potentially lead to reduced drug efficacy. And this is reiterated in the FDA's guidance on drug-drug interaction studies for therapeutic proteins. Here, the FDA recommends that a DDI study is done for small molecules co-administered with the GLP-1 drugs due to the delay in gastric emptying. Alternatively, this could be evaluated with PopPK or PBPK modeling, but nonetheless, the FDA does want to see this type of interaction evaluated. Now, to assess gastric emptying of a GLP-1 drug, typically the paracetamol or acetaminophen absorption effects study is done. Now, as we heard in the last talk, this is not the gold standard. However, it does represent a real-world scenario where patients taking a GLP-1 will likely take paracetamol, for example. And then it also allows us to apply classic DDI study designs. So in this case, it was a crossover study with 15 type 2 diabetes patients given an oral dose of semaglutide or placebo over 12 weeks. The paracetamol was dissolved within yogurt and then consumed within 15 minutes. And blood samples for paracetamol PK exposure were measured over 5 hours. In the PK profile graph here, you can see the conditions of oral semaglutide in blue and placebo in the gray lines. Overall, the Cmax was decreased by 27%, showing this delay in gastric emptying. At the 1 hour mark, the AUC was reduced by 31%, but by 5 hours, this difference was no longer present. And the Tmax was slightly delayed by about a half hour. Now, the delayed gastric emptying is a hallmark for these GLP-1 drugs, and you can see here that across the various GLP-1 types, the Cmax is decreased anywhere from 13 to 56%. Now, the effect could potentially subside at steady state dosing. And overall, there's really minor effects in AUC and the Tmax. So I do want to note that with the acetaminophen, even though there is delayed gastric emptying, no dose adjustment is needed because of the wide therapeutic range. The GLP-1 effect of delayed gastric emptying does not impact gastric pH. In solid lines, we have the liraglutide condition, and in placebo is the dashed lines. And overall, you can see there's minimal differences between the two over a 24-hour period for gastric pH levels. What could determine the likelihood of a drug's PK to be impacted by delayed gastric emptying is its biopharmaceutic classification system category. So as a reminder, class 1, these are drugs that are characterized by high solubility and permeability. They tend to have rapid and complete absorption, and acetaminophen is our main example here. With class 2 drugs, they have low solubility and high permeability. They're further subcharacterized whether they are weak acids, bases, or neutrals. Class 3 drugs, we are talking about those that have high solubility and low permeability. They are defined by having permeability limited absorption, or may even have site-specific absorption. And then last is class 4, which is low solubility, low permeability drugs, and digoxin is a key example here. Now, the anticipated PK effects due to reduced GI motility and delayed gastric emptying for these drug classes, we would anticipate reduced Cmax with a class 1 drug. And indeed, in the previous slide, we did see the reduced Cmax with acetaminophen. Now, with class 2, because of the increased residence time in the stomach, we could have a higher degree of dissolved drug available for absorption. So here, the AUC and Cmax could be increased with a delay in the Tmax. And then with class 3 drugs, it's intestinal permeability is rate limiting. So we would tend to see a reduced Cmax, delayed Tmax. And then finally, class 4 would depend on what is rate limiting, whether it's the solubility or permeability. So here's a summary of the PK changes of common co-administered drugs with the various GLP-1 drug types. In pink, you will see that the AUC is increased. In gray, either the AUC or Cmax are decreased under these specific conditions. Now, I do want to stress that not all of these are considered clinically significant, and no dose adjustments are needed. The one exception is with oral contraceptives co-administered with a tirzepatide. Here, the AUC is reduced by 20%, Cmax by 60%, and then the Tmax is delayed by 4 hours. So this is a clinically relevant scenario, and indeed the drug label does advise that patients switch to a non-hormone contraceptive method or add a barrier method when initiating tirzepatide treatment or during the dose escalation periods. And what about the case of a narrow therapeutic index drug? Well, we have warfarin here as our textbook example. It's an anticoagulant, and you can see here was co-administered with the various GLP-1 receptors. The 2 enantiomers were assessed, and overall there was no change in the AUC. However, the Tmax, the Tmax was delayed by 2 to 7 hours. And then there was a statistical decrease in the Cmax of 19% with lixisenatide and 22% with dulaglutide. The INR, which represents clotting time, also did not change. So overall, even though there are minor differences in the PK, no dose adjustment is needed with warfarin and when co-administered with these GLP-1 drugs. But because it is a narrow therapeutic index drug, it's important to assess this. There are a couple scenarios where there may be a PD drug interaction. For example, sitagliptin is a DPP-4 inhibitor. DPP-4 is the enzyme that degrades GLP-1, and it could potentially increase the GLP-1 exposure, leading to an increased risk of hypoglycemia. Erythromycin is a prokinetic. It's prescribed for constipation and stimulates gastric emptying. So this could counteract the delayed effect of GLP-1 gastric emptying. And then when combined with, say, insulins or sulfonylureas for glucose management, these are known to lower blood glucose levels, so there could be a risk of hypoglycemia here. In general, the GLP-1 drugs are known for gastrointestinal adverse effects, specifically nausea, vomiting, and diarrhea. Shown in the graph here are the severe GI cases that have been reported to the FDA event reporting system database. And the GI cases range anywhere from 13 to 23%. Other common AEs include dizziness, mild tachycardia, headache, and dyspepsia. And the GLP-1 drugs themselves tend to have a low risk of hypoglycemia. They are contraindicated for certain patients, including those with gastrointestinal diseases like gastroparesis or irritable bowel disease, because these conditions already present with a delayed gastric emptying. And there is a black box warning due to the risk of thyroid cancer, as well as warnings related to pancreatitis and diabetic retinopathy. So when it comes to designing a GLP-1 interaction study, there are a few considerations. Here, with the participant selection, you could include either healthy volunteers or overweight or obese participants. The latter really representing the indicated population. And then it's important to account for, say, a 10 to 20% dropout rate due to the prevalence of these GIAEs. especially if the study is going to be a longer term study with multiple dose escalations. So with the drug interaction study, single dose may represent a worst case scenario, whereas at steady state, the effect of delayed gastric emptying could potentially subside. And then to mitigate some of these AI risks, it's important to have rescue medication like antiemetics available for severe nausea and vomiting, and have this listed in the protocol. Another option is to potentially minimize these AEs by providing smaller meals, lower in the fat content. And then it's important to also measure and monitor glucose levels while the participants are in the clinic, and have rescue glucose on hand in case of hypoglycemia. Now, if you're planning to procure a marketed GLP-1 drug, it is getting a little bit easier, but manufacturers are still recovering from drug shortages and the rise in demand. So we suggest to plan ahead. Now, the increase in obesity rate will likely lead to a growing demand and more patients on the GLP-1 therapy. Currently, there's only a handful of clinically relevant GLP-1 drug interactions. And for new drugs in development, new small molecules that will be co-administered with a GLP-1 receptor agonist, here the BCS class could help anticipate the effect of gastric emptying. In some cases, you may need to also evaluate the effect of GLP-1 PK or a PD interaction as well, depending on the study drug mechanism of action. And then for innovator drugs, the extent of delayed gastric emptying should be evaluated. So we would recommend doing the drug interaction studies with acetaminophen, an oral contraceptive, digoxin, warfarin, statin, and an ACE inhibitor. By doing so, you'll cover all of the key BCS classes as well as narrow therapeutic index drugs. And then lastly, I will say expect GI AEs, power the study appropriately, and then mitigate these AEs if possible. So with that, I'd like to thank you all for your attention. We've placed a link here to our product labeling fact sheet that highlights several of these clinical pharmacology study designs that we've discussed today. And I'd be happy to take any questions. Thank you.
Thank you so much, Agonists and Sabina, for this great presentation.
I have a second question for you, Sabina. Are there any other examples of clinical DDI studies with GLP-1 agonists? And is it better to look after the DDI, a single dose of GLP-1 agonists, or at steady state?
Yeah, thank you for that question. So during the talk, I did give a couple examples of DDI studies with GLP-1, like the acetaminophen oral contraceptives, for example. I didn't have time to get into some other examples, like those with metformin or levothyroxine, but overall, they were also not clinically relevant. And in terms of the question, is it better to look at single dose or a steady state? As I was mentioning, the single dose of the GLP-1 could really represent that worst case scenario where you see the maximal effect. With steady state dosing, while you would anticipate or maybe expect that it would be a greater response, what we saw with the acetaminophen was that there was really subsided effects or lower PK changes. And that's because for the GLP-1s and delayed gastric emptying, we tend to see the tachyphylaxis response. So with a new drug in development, say a new novel GLP-1, then I think it might be important to look at both scenarios, both an acute effect and then the steady state as well. So thank you for that question.
Okay, thank you for the wonderful answer.
I have another question for you, Sabina. What implications do the GLP-1 drugs have on perioperative medicine and ensuring an empty stomach before anesthesia?
That's another great question. I am aware that the American Society of Anesthesiologists did put out a consensus document regarding the use of GLP-1s in preoperative surgeries. And this document actually came out last year. And some of the concerns that this group raised is that the delayed gastric emptying induced by the GLP-1s could increase the risk of regurgitation or even pulmonary aspiration of gastric contents during anesthesia or when patients are sedated. So they recommend actually for daily dosing to hold the dose the day before surgery. And if it's a weekly dosing, then at least hold the dose 1 week before surgery. Thank you.
We have another question. This is from Thomas Crow. It says, nice presentation. Is there a consolidated list of clinically significant drug-drug interaction with GLP-1 agonists? And this is to Sabina.
Yes. So as far as I'm aware, I haven't come across a consolidated list. The only clinically significant drug interaction that I was able to find reviewing drug labels and the documents and literature is really that tirzepatide oral contraceptive drug interaction. But I am not aware of a full detailed list.
Okay, thank you. We have another question from Aram Oganesian. He asked, how common is gastric stenosis with GLP-1 agonists that last for a prolonged period? For example, a subject vomiting pizza 10 days ago. We have heard of this.
So, yeah, and that's a great question. I'm not fully aware of the stenosis effect or how long that could last. I think that's something that I would need to really further look into. But sorry, I don't have the answer for you here.
We also have another question from Levi Hooper. She asks, in addition to gastric emptying, how does decreased small intestine motility impact the absorption of orally co-administered drugs? Could increases in small intestine transit time potentially lead to an increase in fraction absorbed and drug exposure?
Yes, I would say definitely that's the case, especially with the BCS2 class of oral drugs where they could be really impacted by the effects of solubility and permeability.
We have another question. Is there a way that we can differentiate whether drug interaction is being driven by changes in gastric emptying versus other drug-drug interaction mechanisms like enzyme or transporter inhibition or induction? Is there potential of drug-drug interaction interplay between changes in gastric emptying and intestinal drug metabolizing enzymes or drug transport?
So I think that's a great question, and I guess it depends on what the two drugs are for the evaluation of a drug-drug interaction. With the GLP-1s, we know that they have this delayed gastric emptying effect. They aren't metabolized by CYPs or really even taken up through the transporter enzymes because they are peptides. So there it's really driven by the delayed gastric emptying. If you are talking about small molecule drug-drug interactions, then for sure PGP, BCRP definitely play a role in that potential drug interaction. And unfortunately, I don't think we've been able to bring our note back to the presentation.
So I have another question for you, Sabina. And what is the time to onset of GI adverse events are there differences in the onset of frequency between oral and sub-Q GLP drugs?
Yes, so the onset of GIAEs is a topic of great interest with these GLP-1 drugs. And from the FDA data reporting system, and then also some pharmacovigilance studies, we know that on average for GLP-1 drugs, the median time to onset could be anywhere within hours to about 24 days of drug dosing. And most patients would experience these GIAEs within the first week of dosing, their initial dose, and then with the dose escalations as well. In terms of oral versus subcutaneous prevalence of GIAEs, overall, they're pretty similar between the two. With the nausea and vomiting, we see them at quite similar rates. The only thing is that with the oral GLP-1 drugs, the median time to onset is a little bit shorter. So typically, I think that it's between hours to 15 days, whereas with the subcutaneous drugs, as I was just saying, it was between 0 to 24 days. So small differences there, yes.
How does a GLP-1 delay gastric emptying? And why was only the tirzepatide DDI clinically relevant?
Yeah, so the GLP-1 delays gastric emptying by affecting the vagoafferent signaling, and it also can impact the gastric smooth muscle cells as well, and reduce the gastric muscle contraction. So that's how it actually plays a role in delayed gastric emptying. Now, like I was saying before, there is like a rapid desensitization of the delayed gastric emptying. So some studies have shown that with a first meal, the delayed gastric emptying with the GLP-1 is much greater than, say, after a second meal. And that's part of that tachyphylaxis, diminishing effect response. And this can also persist with multiple dosing. So that's why at steady state, we could potentially see a less strong effect with the PK. Now, the second part of that question was related to the tirzepatide effect in oral contraceptives. So just a reminder that tirzepatide is a dual GLP-1 GIP receptor agonist. And the GIP itself isn't known to cause delayed gastric emptying, but the GLP binding to the GLP receptor has a quite high affinity. So that's why we would see this really strong effect with the acetaminophen. And if you remember, it was about a 50% decrease. Now, in that oral contraceptive drug-drug interaction, it was a single dose of the tirzepatide. So that's where you would see that largest PK response. And because we saw with the oral contraceptive, not only a decrease in the Cmax, but also the AUC, so the overall exposure, this could potentially lead to reduced efficacy or unplanned pregnancy. So that's why it was deemed clinically relevant. And I think overall, when trying to decide if it's clinically relevant or not, it really depends on the extent of the delayed gastric emptying, as well as the therapeutic index of the co-administered drug itself.
Thank you, Savannah, for these great responses. And so we have another question. You can add an arm to your DDI study evaluating acetaminophen to potentially differentiate the mechanism. Delayed G versus transporter.
So if I understand the question correctly, it's just potentially a comment that the delayed effects could be evaluated with a GLP-1. Is that correct?
Yes, I think, that would potentially mean from what the participant is asking. I think it's a follow-up of additional information, I think. And then we have another question too for you, Savannah. It says, if you already did high fat food and no delayed empty does not affect our drug AUC? Can we predict GLP drug will not affect the AUC?
That's an interesting question. So if you would anticipate a change with the or see a high fat
AUC.
effect, would you really then expect an effect with a GLP-1 receptor agonist? I think potentially yes, but then again, it comes back down to the magnitude of that effect and if it's clinically significant or not. So you could potentially see changes in the PK that may or may not be statistically relevant, but then the clinical significance would depend on the therapeutic window of that drug.
Thank you so much.
Thank you, Sabina. If either of you can answer this question. Do we know that the rationale as to why we see delayed gastric emptying after initial dosing versus limited effects with continued dosing?
Right, so that comes back down to the tachyphylaxis response. So there's a bit of a desensitization with the vagal signaling, and that's been shown with this delayed gastric emptying specifically. In terms of the glucose response and improvement in glucose uptake, insulin sensitization, we don't see a tachyphylaxis with GLP-1s, which is why they are such important and valuable products for diabetes and weight management. Thanks.
Okay, makes sense. Thank you so much, Sabina. Thank you, Sabina and Arnaud, for the wonderful presentation and for your time. And thanks to all the attendees who were present today. And over to you, Lisa. Thank you.
Thank you so much, Archie and Isaac and Sabina, as well as Arnaud. Unfortunately, we weren't able to get him reconnected, but we will follow up with a few questions that were specific for him, and we'll post his typed responses with the recording of today's session. So again, thank you all so much. This is the virtual world we live in. And Sabina, thanks for handling all of the questions today from our audience. We will have this recording posted in the event lobby later today, and then up on the ASCPT online learning library next week. Join us for more great sessions. We've got plenty of them posted on ascpt.org/online learning. Again, thank you to our panelists today and have a great day, everyone.